guest today is Professor Preeya Khanna. She received her PhD in a joint graduate group in bioengineering here at Berkeley and also at UCSF. She completed her postdoctoral training in the Department of Neurology at UCSF. Her research combines sensory motor system neuroscience, network modeling, and neurotechnology development, and those principles can then be used in various types of brain-machine interface therapies, such as neurofeedback and neuromodular approaches for restoring movement control in patients with damaged sensory motor systems, which I think she'll probably talk to us about today. So thank you very much for coming and listening to your talk today. Thanks again.
Sure, thanks. All right, hi everyone. We're a pretty small group, so yeah, feel free to interrupt at any time point with questions or comments. Yeah, make this as casual as we want. So I'm very excited to be here today. As John mentioned, I'm both in the Department of Neuroscience as well as EECS, so I'm really at this interface of kind of thinking about what are the problems in neuroscience and biomedical problems, and then thinking about how to use technology to both study those problems and also design solutions. And so my big picture interest is in developing models of the very commonplace but remarkably fast and flexible dexterous movements that you and I do every day. And then in leveraging these models to try and engineer new types of therapies for people with movement disorders. So just to motivate that problem a little bit more, I'll start just with the patient population that I've been studying the most. Oh, here, let me just get rid of this, which are stroke patients. So stroke is the leading cause of long-term disability here in the US. We have people experiencing about 800,000 strokes per year, with about 7 million stroke survivors in the US. And despite recovery and physical therapy, only about 50% exhibit full recovery, and so 50% remain with disability even after these interventions. And so just to give you an idea of the type of movement impairments that I'm talking about, I'm going to show some data that I collected during my postdoc at UCSF. So this is a person that came in, they had a unilateral stroke, which is damage to one hemisphere of their brain. And that was ended up being their left hemisphere. So their left limb is actually intact. Their right limb is the one that has impairments. So I'll show you, first of all, some data collected from them from their unimpaired limb, doing a task where they reach out, they grab a ball, and then they release that ball. So you're going to see them doing this many times in a row. So this is them just reaching out, grabbing a ball, moving it, releasing it, and repeating 10 times in a row. So you'll see the trial count indicate there. And so you can see this is very fast, repeatable, very flexible. This is probably what you or I would look like doing this task. And so now we can take a look at their affected limb and can see a very different picture. So you can see that first it's considerably slower. There's some difficulty in coordinating the joints. There's some tremor, it looks like. And then there's also just some difficulty in repeatability of the movement. So every one of these trials is just a little bit different from each other. Okay. And so if we take a coronal slice of the brain, we can get a better indication as to why there's so much variability and multimodality in these types of symptoms. So what I'm highlighting here are some of the pathways in the brain that we know are really critical for control of movement. So the purple one is the cortical spinal pathway. This is the canonical pathway that takes signals from the top of your brain down to your spinal cord to animate your muscles. There are also cortico-cortical pathways that do things like integrate goals and sensory feedback into ongoing movements. And then cortical-subcortical pathways, cortical-cortical pathways across hemispheres. So you definitely don't need to remember all of this, but the main point is that there are a lot of different pathways that are important for the smooth, dexterous control of movements that we have every day. And so if we look at where most strokes tend to occur, this is now a map, a stroke density map. And so the higher color, or the warmer color here, indicates a higher probability of participants having a stroke at that particular location. You can see that there's really a big hotspot right at the center of the brain, which is actually right at the intersection of all of these different pathways. And so I hope this gives you a sense as to why individual patients have such different symptomology and why there's such a multimodality of symptoms, why we see slowness and weakness and tremor and coordination problems and learning problems all across different patients. Okay, so this is the problem. And now I'm going to talk a bit about, well, what technology and what tools do we have to address this problem right now? So one class of technologies that, is that okay? Okay. One class of technologies that I'm very optimistic and excited about are what I call neural interface devices, specifically neuromodulation devices. So these are devices that are implanted in the body or put on the surface of the body and inject small amounts of electricity to bias the functioning of the nervous system. So the classic example of this is the deep brain stimulator that was developed in the 1970s for people with Parkinson's disease. So this is implanted deep in the basal ganglia. And injects small amounts of electricity and is actually remarkably able to stop tremors and help stabilize movements in Parkinsonian patients. Since then, though, there's been quite an explosion of technology in this area, both through academic developments as well as companies now. So there are vagal nerve stimulators, there's non-invasive magnetic stimulation, there's peripheral nerve stimulation, like a watch that you can wear that delivers stimulation to peripheral nerves, spinal cord stimulation, even devices that are wearables like headphones that are thought to help with athletic training and movement performance enhancement, and now some even very exciting wirelessly powered devices that are starting to become available. So this is all very exciting, but why are we optimistic about this particular type of technology for upper limb and disability? So one is that they have been used to treat movement disorders before. So not just Parkinson's disease, but essential tremor, dystonias have been treated with these types of devices. And so we know that they can operate on the kind of fast timescales that movement happens on. And the second is that these devices do operate on very fast timescales. So unlike a drug that takes maybe an hour or 2 hours to wash into the system and then has not a fully saturated concentration, even when it starts to wash out, these devices can operate on the scale of milliseconds or hundreds of milliseconds, which is the scale that our movements happen. Third, these devices can be very precisely targeted in a spatial way. So in the same way that I showed you that map before, people have very different types of damage depending on exactly where their stroke occurs. There might be different types of pathways that you want to strengthen or increase excitability of. In some people, other pathways you might want to in other people. And so because these devices can be very precisely targeted, they're very promising for being able to address the individuality that people have in their deficits. And then finally, not only can the spatial targeting be personalized, but also you can personalize the waveforms. So the way that the electricity is actually delivered into the system, the patterning it gives, the timing of that patterning, that can all be very personalized as well. And so that's why I'm really excited about this class of technologies for being able to address this particular problem. So in concert with being able to deliver electricity to the system, there are also a host of technologies that have been designed to be able to sense from the system. So sense the ongoing state of the participant. We can do that through a variety of different neural recording technologies. So here, this is just a schematic of non-invasive to fully invasive types of recording from the nervous system. And then these are some of the FDA-approved devices that are used to actually record from the nervous system now. So Neuralink's device, I shouldn't say it's not quite FDA approved yet. It's in investigational use right now, but that's largely thought of as the state of the art for being able to record from many, many neurons inside the brain, very high density. BlackRock Neurotech, that's actually is the FDA approved device that's been used for a lot of the brain machine interface work. So these are devices that are implanted in the brain and can record from large populations of neurons. But beyond neural recordings, there are also now a host of other devices that help us monitor the state of a person. So Control Labs has a very exciting high-density EMG, surface EMG wristband. So that's something that we could use potentially to detect, is someone about to move or trying to move? How might that influence what sort of stimulation we give to the system? There are also, for example, computer vision systems that can give us, the state of a person more in pose space. So what is their posture? Are they about to initiate a movement? Are they trying to, play a sport? Or are they just sitting on their couch? And then even kind of longer term tracking, like physiology tracking. Is someone kind of in a good mood? Is someone sleeping well? What is the general physiology of the state? So all of this technology is also very exciting for trying to figure out how might we be more intelligent about delivering this type of technology, the neuromodulation technology. So despite all of this technology that has emerged largely in the last couple of decades, right now there's no device that's part of standard clinical care for upper limb disability. And so this is the problem that I really want to go after in my group and moving forward in this talk too is what I'll be talking about. So before I get there, I just want to take you through a vision of what I think the future of upper limb disability treatment could look like if we have a better understanding of a couple of things. So imagine that someone has a brain injury. They come into the hospital to have that brain injury treated. What I would love to be able to see is that that person has an array of tests that are done on them, not just neuroimaging tests, not just their patient history, but physiology measurements. So what sorts of neural signals do they exhibit when they try to move? What sorts of physiology and muscle signals does it look like when they try to do some of these coordinated movements? And then based on that, we get a better understanding what systems are actually intact, what pathways are intact, and which ones are damaged. And so from that, we can actually develop a personalized prescription for that particular person. And so that personalized prescription can choose from a buffet of different devices. So maybe, for example, that person mostly has movement control intact, but they're just very weak. And so maybe we would want to support them with something like a wearable robotic system that just gives them a little bit more strength against gravity as they're trying to move. Or maybe it's someone who has a tremor, and so they really can move quite effectively, but they just need their movement stabilized. And so maybe something like a peripheral nerve stimulation device is something that could be prescribed. Or maybe more broadly, they can initiate movement, but they really can't coordinate movement very well. And so that might be appropriate for something like a central nervous system stimulation device. And so I think this sort of personalized device prescription can be something we really get at if we understand two main things. So the first is, we really need to understand, what does normal physiology look like? So what should the brain be doing during movement? What should muscles be doing? How do pathways actually coordinate to generate movement? And so this is kind of going to be the first topic I'll be talking about, which is advancing a systems neuroscience understanding of fast, successful movements. And then second is, when we do develop these devices, how do they actually interact with that system? So when I do something like stimulate the peripheral nerves or stimulate the central nervous system, how does that actually affect the physiology and thus affect the way that the brain controls movement? And so these are going to be the two topics that I'll be talking about going forward today. So first, we'll start with advancing models. So before we get to how the brain controls movement, let's take a step back and try and think about the behavior itself. So if we go back to this video that we started with, this is the affected limb. What actually specifically makes this behavior ineffective? And so we said, or I said, a few different adjectives. So I said that the behavior was slow. I said it was tremorous. I said, there's maybe difficulty in coordination. But let's ask a clinician. So how do actually clinicians right now assess whether this behavior is effective or not? So what they do is, there are a variety of different behavioral clinical scales that can be assessed. So often these are scales on a scale from 0 to, for example, the Action Research Arm Test is a scale from 0 to 57. So if you're a 0, you're paralyzed. If you're a 57, you can move just like you or I. And so there's a bunch of different subcategories where you do these different tests like gripping and grasping, putting your hands up, and timed trials for different behaviors. And then all of that information gets compressed into one final score you get at the end. And so this particular person that I showed you is a 34. And so that means that they're not paralyzed, but it also means that they do have some significant movement impairment. So they're not moving just like you or I. And so this is currently the state of how movement impairments are assessed. So this compression is very useful in some ways. It's a very fast way to figure out what the impairment is of a person, but it does miss a lot of the details. So it misses a lot of these adjectives that we described: the slowness, the tremor, the lack of coordination. It also misses, where is the problem? Is it an arm problem? Is it a wrist problem? Is it a finger problem? And so these are also things that we're missing through this lower dimensional description. And so one of the projects that I worked on as a postdoc was developing a wearable technology. So this is a wearable glove with electromagnetic sensors. And we put this on people as they were doing different movement tasks. And this is actually the glove that we used to collect the data in the video that I just showed you. And so we had people doing these reach to grasp tasks. And the thing that I want to show you is that we actually were able to recruit 3 participants that had stroke that actually all had very similar ARAT scores. So that was that clinical score on a scale of 0 to 57. So I know this is a little bit small, but this is a person that has a 38 at the top here. Let me get my cursor. A 38, a 34, and a 36. And these people all have subcortical stroke. So this is actually how stroke is often characterized, as either cortical or subcortical, not even like where in the cortex or subcortex. And so, from a clinical perspective, these are all very similar patients. Now, what we're going to do is we're going to take a look at what their individual joints are doing during these movements, these reach to grasp movements. So using our glove, we could focus, for example, at the thumb IP joint. So that's this thumb joint right here. And so what I'm going to be showing you is a few plots that look like this. The x-axis is time, which has been warped to just be % of the time through the trial as people are reaching out, grabbing, picking up an object, and putting it back down. And then the y-axis are the change in degrees. So it's how many degrees of flexion this thumb joint is undergoing. So for this participant at the top here, you can see that, so the purple is the participant. The black is going to be a neurologically intact person, so just someone from our lab who did this task. And then each of the individual lines are one trial. So you're going to see 10 trials in purple and 10 trials in black. So what you can see here is that for this participant, when they did the task 10 times, sometimes they were very successful at being able to flex their joint. Other times, though, it's a flat line. They weren't able to flex their joint. You can see in the neurologically intact participant that they're always very consistently able to flex this joint. So this participant sometimes is able to do it, sometimes not. This participant actually looks quite different. So there's quite a bit of noise or tremor in this participant, and there isn't much modulation above that tremor. So we would consider this as quite a bit of impairment where they really can't bend this joint very much, but it's dominated by the tremor. This participant's a little bit different. So there's more consistency on trial to trial. So all of the purple traces look pretty similar to one another, but the amount of flexion is much less than what you see in the neurologically intact participant. Okay, so that's the thumb joint. So let's take a look at another joint. So this is elbow flexion and extension. So here, there's actually quite a bit of similarity across these participants. And so this again is the neurologically intact participant in black who's extending all the way, grabbing this object. Here you can see that in all these participants, they just can't extend nearly as much as the neurologically intact participant. And then finally, one other joint we'll look at is the shoulder add and abduction. So this is this joint here. And so what you can see is in this participant here, this green trace actually extends even further than what the neurologically intact participant does. And so this is potentially evidence of some amount of compensation. So perhaps they can't extend their joint as much, and the way they're compensating for that is overextending their shoulder joint. So here we see overextension in this participant. Here again, we see almost no modulation, just dominated by tremor. And here in this participant, we actually see pretty normal shoulder add and abduction. Okay, so why did I take you through this? The main point is that even though clinically these people all have subcortical stroke, they all have very similar clinical scores, they have very different patterns of joint dysfunction and very different patterns of the way that they're actually doing this movement. And so what I would argue is that we really need to understand what different pathways contribute to these different types of dysfunction. Is it, for example, that this participant has a slightly different neural pathway that's damaged compared to this participant versus this participant? And if so, we need to better understand that, because if we want to design therapeutics that are specific for each people, we need to know what are their actual dysfunctions. And so in order to do that, we need to understand both the behavioral side and then also the corresponding neural side. And so I would argue, too, that this is not just a neuroimaging problem. It's not just that we need better, for example, MRI scanners so we can see the details of these different patterns of dysfunction in the brain. Because one thing that we know is that after a brain injury, the brain has this marvelous mechanism of recovering and strengthening existing connections that are still there. And so it's not just that we need to see what was damaged, we also need to see what was strengthened through physiology measurements and through these sorts of behavioral measurements. And so towards that, one thing that my lab is working on now are technologies for better quantifying behavioral assessments. So we're really interested in trying to understand what is the distribution of normal joint movements that we see in everyday people, and then how does that compare to what we see in people who have impairments. And so one thing that we're trying to do right now is use computer vision post-tracking methods to better characterize and build data sets of neurologically intact people at different ages, and then compare that to people who have impairments and see what joints for them are out of distribution versus which ones are more normal. And can we kind of identify subgroups of people based on that? And then a second approach that we're using is actually building a wearable technologies. And so we have some folks in the lab that are working on these silicone-based strain sensors that go across joints, like what you're seeing up there. And these are filled with liquid metal channels. And so when the strain of the silicone sensor changes, as someone bends their joint, the resistance of this channel changes. And so we can measure that to actually measure joint angles. And so our hope is that these are something that is our vision for this is that we have these strain sensors on these different parts of on the hand. We're able to measure kind of hand opening and closing. We augment this with some inertial measurement units that we have on a wristwatch and then the palm here, so we can get kind of wrist flexion and add and abduction and pronation and supination. And so then all together, we're hoping to build this sort of multimodal sensor device that's very easy for people to put on and take off, and they can wear about their everyday life. And so some of the things that we're really excited about are just collecting larger data sets of people as they're going about their activities of everyday living. We're interested in questions like, if you put this on during physical therapy and are practicing certain movements, how well do those generalize outside of physical therapy? And then more broadly, just building up these data sets to try and understand what types of joint movements are abnormal. And so we're at early stages of this, of actually developing the sensors in the lab. This is just the process for what these look like and what some of the sensors look like on the skin. But more broadly, we're hoping to be able to use a combination of both these computer vision and wearable approaches to get at these sorts of questions. All right, so if we think about advancing models, our first thing is we're really interested in having models of successful behavior in patients. We want to know what aspects of their behavior specifically are impaired, what aspects are successful. And so we think we can do this with wearables and better data collection in people and neurologically intact as well as in patients. All right, so now I'm going to move on to now how does the brain control these movements? So if we have a good model of what sorts of movements are impaired and what sorts of movements are actually regular, can we do some inference as to how does the brain actually control good movements? And so here I'm going to be talking mostly about recordings that are from inside the brain. So I'll use this phrase single units or single neurons, and that refers to signals that are recorded from inside the brain that have been high-pass filtered to identify putatively spiking neurons. Can talk more about how one does this in the future, or if anyone has questions, but just wanted to clarify that terminology first. Okay, sorry. Let me actually skip through this. That shouldn't have been there. Here. Okay, so when I started my postdoc, a question that I was really interested in is, I mentioned before that the brain has this miraculous capability of when it undergoes a neural injury, it can actually recover spontaneously to a large degree from that. So we saw that in stroke patients that spontaneous recovery and it happens in about 50% of people, whereas 50% are left with some sort of impairment. And so what I thought is if we can better understand how does the nervous system do that recovery spontaneously, then we can start to design types of technologies that push the system towards that more recovered state. So first we needed to characterize what that natural recovery looked like first. And so we did this in a preclinical model. So my lab works with rhesus macaque animals. We train them to do reach to grasp tasks. So they reach out, they grab a pellet, and then they eat it. And they're very tasty, so they're very motivated to do this. And so then what we do is we created a small lesion in the motor cortex of the brain. So this is a part of the brain that controls movement of the hand and the arm. And so that created a very temporary deficit in these animals. And so what we saw is that, for example, when they did these reach to grasp movements, so here I'm plotting many trials of their kinematics on top of each other. Initially in the impaired state, they were quite variable, similar to that video that you saw from UCSF. But after some time, they became, and this is after about a month or so, they became much more stereotyped again. So we could hardly tell that they had had an impairment at all. And so what we were very interested in doing is studying the change in the brain activity between this time where they were impaired and recovered to better understand what was happening to enable this control. So as I illustrated here, we had intracortical recordings from a part of the brain that I'll call PLC, or paralesional cortex. So it just means next to the lesion in cortex. And this was actually a brain area that we know from other work is causally related to the animal being able to exhibit improved control after the brain injury. So we know that this area takes over control, basically. All right. In there. Okay. So these are an example of some of the single neurons that we were able to record from this. So I'm just going to put a note here that with this technology, we're able to get about 30 to 40 simultaneously recorded single neurons. And some of my future work, which I'll describe in a couple minutes, we're going to be able to increase that by almost an order of magnitude. And so take note that we're at a time in neuroscience where we're about to learn a lot more about the brain, just given the technology we have. But given what we had at this point, we were able to record some of these units. So for each of these is a different electrode that's at a different point in this paralesional cortex. The x-axis here is on the scale of about 1 to 2 milliseconds. And so these are actually the single unit action potentials from your basic neuroscience course, the spiking of the individual neuron. So what we're able to do is look at each of these individual neurons and see how they modulate during behavior. So here the x-axis is time. Time at 0 is the movement onset, so when the animal starts to reach. And then each of these rows here is a different trial. So here they're doing, looks like about 30 to 40 repetitions of this behavior. And what the dark regions indicate is when that neuron fires a lot. So we can see that this neuron really prefers to fire kind of before the animal reaches and right around movement onset, and then is very quiet afterwards. So another view of this data is to average over the y-axis. And so we get an activity trace that looks like this, where again, x-axis is time. Now higher up indicates more activity of the neuron, lower down indicates less activity of the neuron. And so this is a view of the trial average of a neuron. And then finally, we can do what I will be calling a trial average template, where we look at the activity of all of these units at one time, trial average. So again, here the x-axis is time. This dotted white line is movement onset. Each row here is a different neuron. So each of these different observations contributes to this here. And then the color, when it's more yellow, is more activity of the neuron. When it's more blue, less activity of the neuron. Okay, so now that we have these templates, what can we do with them? So the first thing that we asked is how consistent are these templates on a trial-to-trial basis? So if you remember early on in the both UCSF video and in the behavioral plot, I showed that there was more variability in the behavior when the animals were impaired and a lot more stereotypy when they were less impaired. So we were very interested in seeing whether this actually showed up in the brain activity as well. So this is a trial average template when the animals had some impairment. This is a trial average template when they were fully recovered. And what we did is we wanted to compare them to the individual trials that happened on those same days. So one thing that you can notice is this trial average template early on, maybe some of the most salient features are that it's quite blue here, and then there's big yellow bursts around here. But if we look at individual trials, we see that these individual trials are actually really different. So the patterns here don't totally match the trial average. Whereas if you look at the fully recovered state, you can see maybe the salient features are that there's kind of this cascade of yellow activity here. You actually see that really nicely in the single trials here. It's a little bit noisier, but you do see it. And so one of the things that we characterize is how well does the single trial activity match the trial average template. And what we found is that this consistency does emerge as animals recover. So as their behavior gets better, which is lower on the x-axis, this template matching gets higher. And so the main takeaway that we found was that we actually see very good consistent spatiotemporal spiking as animals get better. So a lot of consistency from trial to trial. So this is just the consistency. We can look at some of the other features of the spatiotemporal patterns as well. So one thing you might notice that I kind of alluded to before is that the bursting of activity that happens after movement onset is a bit different between impairment as well as the recovered state. So one of the things I noticed is that, yeah, there's just kind of one big burst of activity in the impaired state, but there's this really nice cascade of activity in the recovered state. So it's kind of burst, burst, burst. And so we can quantify that using a metric called dimensionality, and we do indeed see higher dimensionality of these spiking patterns with recovery. And then finally, one other metric that we like to look at in neuroscience analysis is temporal consistency. So given that you know where you are at one particular time point, using the state of the vector of neural activity, how predictable is that of where you will be at the next time point? With the idea being that a neural network that is very stable and has very reliable connectivity from one time point to the next is going to provide more reliable patterns for control of movement. And so we use just a simple linear dynamics model to fit activity at one time point to see how predictive it is at the next time point. And we find indeed that with recovery, we see much higher predictability of temporal activity. So not just trial to trial is there more consistency, but even time point to time point, there's a lot more predictability. Okay, so all of this to say is that we've just created a model based on data analysis of what happens as the brain naturally recovers from movement. And so this gives us a model of where we'll eventually get to with engineering therapies. So I'll just point out that right now this is really very heavily motor cortex focused. And so this is really focusing on the connections from motor cortex down to the spinal cord. So one of the things that our lab is really interested in doing going forward is trying to elucidate some of the other pathways that are really important for control of movement. And so we're going to be very focused on cortical-cortical connectivity as well as subcortical-cortical connectivity. And so specifically, we know that for control of dexterous hand function, it's not just motor cortex that's critical for sending commands to the output, but somatosensation and the sense of touch is also very critical for how we process and update movement in real time. And so just to show you a very concrete example of this, this is an example of someone who is trying to light a match here on the left. So I'll just show you a video of that. Okay, great. So their job is to pick up a match from here, strike it on a box, and light it. And so you can see that they're able to do that very successfully. And now what the experimenters did was they then numbed the participants' fingers and they asked them to try exactly the same thing again. So even though this person's motor control is intact, they can see their hands, they can't feel anymore. And so here's an example of what their behavior looks like during that. You can see they're fumbling with the match, they dropped that one. Okay, gave up on that one, next match. And then like very awkward posture in the hand. Many attempts at trying to strike it. And there we go, we finally got it. But you can see it took much longer and was a lot clumsier. And so, yeah, I don't think this is surprising to folks who live in cold environments. When your hands are numb, it's really hard to do things like take your gloves on, open doorknobs. But we currently, our best understanding of how the brain controls movement really comes from motor cortex itself. And so we're really excited to try and better understand the coordination between both somatosensory and motor cortical areas in order in these dexterous movements. And so towards this, we're developing a behavior that both people as well as animals are going to be doing. They're manipulating an object through an unseen environment. And then just to come back to more the technology side, previously what we were able to do with our technology was record from these 30 to 40 simultaneously recorded neurons from a single area. And this was using technology that was developed out of Duke University in 2003. So these are these microelectrode arrays that have kind of about 60, so 8 by 8 arrays of individual wires. Each wire has one contact site. You implant these in the brain, as you can see here. But what's been really exciting in the last, let's say, 5 years has been kind of the generalization of some of the CMOS processes to make these much higher density, addressable, high density neural probes. And so this is the neural pixel probe that is being developed by IMAC right now. So this is a probe that now has up to 4,400 sites on a single shank. And you're able to record from 384 of those sites simultaneously. And so this is a huge game changer within the neuroscience field. One of these can be implanted in one area, a second one can be implanted in a second area. These are kind of the traces that you're able to get. So this is 5 milliseconds. These are 384 of these channels. And you can see already very rich activity in kind of these single unit action potential spikes occurring over the geometry of the array. So these are kind of maybe one spike here, one here, one here. But mind you, this is just 5 milliseconds. So if you recorded for hours and hours, you would get much richer activity. And then finally, one thing I'm really excited about is that these probes record such high density that the probability of recording neurons that are putatively connected or synaptically connected is much higher now. And so previously, when you had 30 or 40 neurons, like the probability of any of those neurons being connected in a statistical way that you could infer from data was very low. It's close to 0.1%. But when you have so many of these neurons, that probability increases. And so this is actually a map of kind of putative synaptic connections on a single probe. And so when we have these arrays in 2 different locations, we can then actually identify pairs of neurons that are talking to each other and then specifically analyze those particular pieces. And so this is just kind of, again, technology is really enabling us to understand much more about how the brain controls these types of complex movements going forward. So that was just a tangent on our future work. So now I'm going to direct us more towards the engineering therapy side. So we built up a model, or we'll hopefully be building models of behavior. We've built a model of how the brain controls movement when it recovers from a brain injury. So now how can we use our understanding of this to actually develop sort of therapeutics that we think might help improve dexterity and function? So I showed you this previously. This is one of the templates, the spatiotemporal templates. So again, one of the features we found very saliently across all the animals that we did this study in was that there were these sequential bursts of activity. So one burst, then the next, and then the next. And so one thing we did was we created a very simple neural network model of just neural activity bursting from one time point to the next to the next. So here, this network model contains an input, then it triggers activity at the next pole, then the next pole, then the next pole. And so what that looks like in time is this. You get a pattern that's very simple, just like this. And so we made this model to actually recapitulate the features that we saw in the data here. We then created an impaired version of the model to create kind of the early stage. So this is the impaired model. This is the recovered model. And these are just single trials here. So if you did the impaired model and had many trials of it, this activity pattern would actually die out at different time points. And then we did exactly the same data analysis that we did in the real data now on our model data. And so we found basically that the template match was higher in the recovered model, the dimensionality was higher in the recovered model, the temporal predictability was higher in the recovered model. So now we have a model of the impaired network and the recovered network. And so we get to our big question, which is, okay, we know how the recovered system looks like. We know what the impaired system looks like. Is there a way we can think of injecting electricity or designing electrical current to make the impaired system look more like the recovered system? And so part of the answer to this came from some of the work that was going on in my postdoc lab at the time. So there was a type of electrical stimulation where we placed electrodes just on the surface of the brain and provided very weak electricity passing between these electrodes. And so the idea here was that if you looked at neural activity that was recorded when there was no stimulation on, the activity was very random. There wasn't a lot of structure to it. But if you looked at activity when there was stimulation on, when we turned on this very weak current, we started to see much higher coherence in the spiking activity. So it was almost though there were these excitable phases of activity and neurons were coordinating to spike around those excitable phases. And so one detail of the model that became very interesting to us was that if you look at the input part of the model, so here, this like red part where input neurons are triggering the activity of the model, if that activity is much more spread out, you tend to get activity patterns that aren't as successful at propagating all the way through the network. Whereas if that activity is very coherent and very tight, you actually get very good propagation of activity through the network itself. And so we were very excited about this type of stimulation that improved the coherence of activity, because we thought if we could align the stimulation to the beginning of movement, we might be able to create these inputs that were a lot more coherent and could give rise to activity patterns that propagated better. And so again, we were able to model this. So this was no stimulation. This is if we injected an alternating current stimulation. And so you can see that this alternating current stimulation creates these bands of activity. So again, these pockets of excitability and inhibitory activity. And when we align the excitable parts to the beginning of the trial, we actually get much better propagation in the network model itself. And so we actually did this test in our animal. So we had our lesioned animals with the array implanted. We implanted these electrodes on the surface of the brain. And then we passed the same alternating current stimulation that was very successful in our model into the brain itself. And so this ended up creating these time-varying electric fields in the brain that mostly were strong at the peak as well as the trough. So you can think of this as basically a time-varying voltage gradient that goes in one direction and then the other direction as the sine wave flips from peak to trough. And so the main thing is, does this actually work? And so here I'm going to show you a video of the animal who has an impairment reaching out and grabbing a pellet with no stimulation on. So the pellet is right here. And so you'll see their hand attempting to grab the pellet. So let me just play that first. You see they have some difficulty, but they do get it out. And then this is the same animal on the same day doing exactly the same task, but now the stimulation is on. And so again, the pellet is right here. So the top video will play just for reference, but I encourage you to look at the bottom video. And so you can see that they're a lot faster. And so this is just one video and one animal, but we repeated this over many sessions in animals with actually slightly different lesions as well. And we did find that indeed, yeah, we were successful at improving grasping performance. And so if we think about engineering therapies, what we were able to do is basically create this model that was based on our data analysis of the impaired network and the recovered network. And we found stimulation parameters that were actually able to enhance the propagation of neural activity throughout that model. And we found that that actually generalized quite well to the not awake, but in vivo case, where animals showed better grasping performance as we delivered stimulation. And so, yeah, I'd be happy to, there's a lot more to this as to why we think this is working, what exactly about the stimulation parameters were successful versus what types of stimulation we think might not be successful. But the exciting part is that we were actually able to rescue grasping performance. Empirically, that's something that's been very difficult in the clinical population. And so, yeah, just a couple of details about the stimulation itself. So again, this is no stimulation. We did find that it was important for the stimulation phases to be properly aligned to the behavior. So if we had these excitable phases kind of not well aligned, we actually didn't see the improvement in behavior. But whereas when we did align them, we were able to see that this pattern actually was rescued all the way. And so this gave us some ideas for how to optimize the stimulation further. So this is an example of an optimized waveform where we took this very low frequency component that was helpful and excitable and was able to help boost neural activity and improve coherence. And we coupled it with a much higher frequency phase that basically just shortened the inhibitory part of that waveform. And so this is an example of a pattern that initially is not properly aligned, and so it doesn't propagate all the way. But if we align that to our new optimized waveform in exactly the same way, then we actually see rescued performance there. And so this is really nice because we think there are actually ways to optimize this stimulation paradigm even further. And then second, in our lab right now, one thing that we're really excited about is not only just delivering open loop stimulation that we kind of synchronize brain activity I'll say willy-nilly, like regardless of what the underlying network is doing. But we're really excited now about trying to detect neural activity in one area and then use the same style of very low intensity stimulation to enhance communication between one area and the next. And so the motivation for this could be that a lot of these strokes actually tend to occur in white matter areas where you're losing connections between cortico-cortical networks. And so if you can imagine that you record from an upstream area, say, a prefrontal cortex area that's giving information about what to do to a downstream area like a motor cortex area that's then figuring out how to do it. If you can improve that communication, you may be able to have better, smoother movements. And so this is a tool that we're developing right now, using algorithmic approaches to try and cancel stimulation artifacts. And then also we're interested in building circuits to try and actually take this exact signal from area 1 and play it into area 2 to boost communication between those areas. The last thing I'll say about the stimulation is that even though this paradigm has many optimizations that could be done in the future, like the waveform, making it closed loop, right now there is actually a company that rolled out of Berkeley that has developed a type of non-invasive stimulation that is based on TMS. So TMS is transcranial magnetic stimulation. It's non-invasive. It basically has a giant coil. It induces a magnetic field that passes through the skull very easily, and that creates voltage gradients within the brain. So TMS is FDA approved for treating depression when it targets prefrontal cortex areas. So lots of people use TMS. And this is now a new type of TMS where you can not only just give these big pulses of TMS to the brain, but you can actually embed them into these smooth waveforms. So similar to this 3 Hertz types of stimulation that we were doing. So this group has now been doing a clinical trial with stroke patients to try and see if 3 Hertz KTMP, which is the name of their TMS type of stimulation, actually improves participants' motor control. So they have people come in, they take a baseline, they do what's called the Fugel-Meyer, which is another one of these clinical scales that give you a number from, I believe this one is 0 to 65. Then they have the people come back 4 weeks later, they take another one. Then they have them do interventions where they have this KTMP stimulation ongoing while they're doing physical therapy. So while they're doing motor tasks, and then they actually do assessment afterwards and a follow-up. And so they've recruited right now some amount of participants, 1, 2, 3, 4, 5, 6 participants. And this is what they're seeing in the Fugel-Meyer scores. So they call baseline 0. So they kind of normalize for each participant's impairment. Then this is what they see pre-KTMP. So this is kind of the natural variability you see when you do these scores over and over. And when they do post-KTMP, they actually see a significant improvement of about +7. And that seems to sustain at follow-up largely, although there is some drop-off there. So one caveat is that these results aren't yet sham controlled. So in all of these types of studies, you want to have people undergo the same intervention without KTMP, but think they're having KTMP just to see how much of this is placebo. But the fact that a lot of it persists after follow-up is very encouraging. And then one other thing that's really nice about this study is they're actually doing motor kinematics as well. So they're not just measuring these scores, but they're also seeing how is KTMP actually making these controlled better. And so this is an example of someone, one of their participants, the DIP joint, I believe it's for the index finger. So the DIP is the very end here. So they saw quite a bit of variability early on, and now post-KTMP, they're seeing a lot more consistency and a lot more extent. So an increased range of motion as well. And so this is very exciting for us because this is suggesting that some of this modeling and mechanisms work that we do preclinically has actually resulted in a clinical trial that seems promising thus far. So future work definitely includes optimizing the waveforms, making this sort of technology closed loop, and then also designing, even though this study was invasive, if there are ways that we can develop tools that are non-invasive that have similar effects, then that's the best because that's the most accessible type of therapeutic for everyone. All right, so now I'm just going to close by talking about one last project. Right now, all of what I've talked about so far is this notion that we can study how the brain recovers from movement, understand what a good model of how the brain controls movement is, and then design therapies to really try and restore that. And I think what you probably saw in the KTMP example is that a lot of times these therapies actually occur in concert with physical therapy. So an intervention like brain stimulation or nerve stimulation, that's often done while people are doing physical therapy because it's thought that it might not only help boost performance during the physical therapy itself, but might boost performance in a way that's sustainable past the physical therapy. So it's kind of boosting plasticity within the brain. But one of the things that's challenging is that there aren't always people that are able to produce movement and participate in physical therapy if they're severely impaired. And so one of the things I think it's important to remember is for any therapy, what are the assumptions behind the therapy? So here, I went through this quite quickly, but we built the model built on the assumption that the main thing that we had to do is just allow this pattern to propagate further. And so the model assumption is that the neural network is actually movement capable, like there's a connection from the brain to the spinal cord and muscles. And if only we can make the pattern in the brain propagate more reliably, then the muscles will follow suit. And so that's one assumption. Another assumption is that the right neural pattern for a given movement has already been formed. So we don't need to relearn how to do the movement. We already know that these are the right neurons to recruit in this order. A third assumption is that as long as we can make this activity pattern propagate all the way, that will yield fast, smooth movements. And so these assumptions are true for what I would call minor to moderate stroke patients. So about 70% of people with upper limb disability are in this minor to moderate category. And so this is very good. But there is a whole another class of patients that are what I would call severely impaired stroke patients. So these are people that are almost on the much closer to the paralysis side than the movement control side. So for them, these assumptions are more questionable. So it's likely that the network is not movement capable. There's some impairment from the brain to the periphery itself. We have no idea if the right neural patterns for a given movement are already formed because these people aren't moving regularly. And it seems unlikely that the main problem is that the cortical patterns just need to be completed and then all of a sudden these people will magically move. And so there are examples of that. So this is an example of an MRI for a participant who is in this category of severely impaired. So this is the lesion here. And so even though they can't move, they're, the very common posture that you might see walking around the street in someone who's severely impaired is kind of arm super flexed, wrist flexed, just kind of like some difficulty, like rotated inwards, and very difficult to move. So this is a participant like that. But even so, a lot of these people, even though they're severely impaired, do still have some connection from the brain to the spinal cord. And so what's illustrated here in red and green are tracings which have gone from the spinal cord all the way up to the brain to show where there's still intact connectivity between the two. And so this green here is evidence that there still is connectivity from the brain to the spinal cord. And so, given what we've learned thus far about what is the good model of control of movement, are there principles we can use to try to develop therapies for these types of people? So one idea that's emerged is that maybe we can give people feedback of their neural patterns, because right now they don't get any feedback about what their brain is doing because they can't actually move. So they don't know, like, was that a good neural pattern to try to move, because they can't actually see any evidence of that. And so one of the features I didn't spend very much time talking about is this local field potential feature. The quick version is that feature tends to be very prominent when you're not moving or when you're resting, and then goes away when you're actually moving or attempting to move. And so this is a feature that folks have taken advantage of to build brain-machine interfaces for rehabilitation and therapeutics. And so this is an example of a participant that's having their neural signals recorded out, and then they can't move, but when they detect that this beta power goes away in that attempted movement state, that actually drives an exoskeleton that's attached to their hand to kind of give them feedback of that movement. And so this type of technology has actually been shown to be successful. People who have undergone therapy with this technology do show some amount of clinical score improvements. And so it's given people almost the ability to get a little bit of movement back. And so one project that I've been involved in is how do we actually bring this to the next level? So one thing that's actually very exciting about these participants is even though they don't have the ability to move, often they have the ability to have these subthreshold muscle activations. So you can ask them to move and you can actually record muscle activity from the surface and see activations there, even if it's not strong enough to actually animate the musculature. The skeletal system. And so one of the things that we decided to do is develop a new type of brain-machine interface that involved both the brain as well as the muscle. So we had invasive neural recordings from the part of the brain that still had the connectivity to the spinal cord. And what we did is we gave that participant, we hooked that up into a brain-machine interface, and we gave participants basically feedback of their neural signals. So as they would imagine moving, that would move an exoskeleton that controlled their own arm. And the idea here is that we wanted to reinforce trial-to-trial consistency in these neural patterns. Then, when we were convinced that people were able to do that well, we then put on surface muscle activity, and we saw what the subthreshold muscle activity looked like. And so we then introduced that into the decoder with the idea that we knew that the person had really good neural patterns for movement at that time, and then we wanted to kind of combine those with neural activity patterns in the muscles that are what I'll call biomimetic. So we have a good understanding of what muscles actually need to activate to move the arm in certain ways. And so we developed a decoder based on that to say, okay, we want to make sure that they're activating their muscles in a realistic way at the same time as activating these neural patterns. And the idea here is that if they practice with this paradigm for a very long time, that we could actually induce these neural patterns to start to control the muscle activity more effectively. And so just for time, I'll show that we were able to do this. And so this is a very long-term trial. This is ongoing for 4 years. We have seen improvement in clinical scores. we have seen improvement in functional scores as well, more so than anyone's really ever seen for this type of participant with this severe of an injury. And so the main takeaway here that I want to emphasize is that we were able to develop this brain-machine interface intervention. We're still working on trying to understand what aspect of that intervention was the most successful at helping this person rehabilitate. But the main thing that we're excited about is that for people that are this severely impaired, if we can just give them access to very long duration, high intensity therapy, we think a lot of people can benefit from that. And so there's evidence that, for example, we can use wearable devices. So, backing up a step, this sort of intervention was something that was a Herculean effort, 4 years of like physical therapy, plus neurology, plus clinical scales, plus researchers all working together to make this kind of brain-machine interface robot something that was viable and something that was fixed every time it broke. That was something that was very time and cost intensive. But if we can actually build wearable devices that allow people to do this intensity of therapy in their homes by themselves, that's something we think can actually be a lot more accessible and potentially have a much higher impact. And so there are examples of some studies that have been able to show, for example, if these are muscle activations before a kind of at-home intervention, these are sorts of muscle activations that you can get with biofeedback therapy after interventions at home, where people are wearing muscle sensors and playing kind of EMG types of games. So all of that to say is that we're very interested in building wearable muscle sensors that allow people to do these sorts of biofeedback therapies kind of in a more accessible way. These are some of the systems that have been published thus far. So a lot of the pain points are that you have signal variability across sessions. Sensors are bulky. They're not very user-friendly. And then people have to set all of this up on their own. That's kind of difficult. And so we've been working with some of the groups here in EECS to build kind of thinner, breathable, flexible, longer duration sensors, such as the one pictured up at the top here, that we think could be better for these types of at-home applications. And so future work will be kind of trying to basically bring the success of this invasive, very high intensity approach to people in their own homes. All right, so I'll wrap up in 1 minute. And so, just kind of to bring us back to where we started, there's all of this technology and neuromodulation and sensing. This is our vision of, where we want to be going forward. And so what I'm really excited about is that I think, by studying the nervous system through some of the examples of studies that I showed you here, using this technology, using even newer technology than I'm showing here, like those high density neuropixels, I think we can really understand what some of this physiology should look like and start to understand in patients what sorts of physiology are they missing? Are they missing reliable neural patterns? Are they missing high dimensional patterns? Are they missing kind of an initiation signal? What are they missing when they come in and have an injury? And through that, we hope that we can actually be more selective about what types of people are getting what types of interventions that are specific for the pathways that are damaged to them. Plus hopefully building new types of interventions as well. So with that, I'd like to thank my postdoc lab at UCSF, my PhD lab that was here at Berkeley, actually, the folks who worked on the ISMOR interventional trial, which I went through very quickly, and then finally my group here at Berkeley in Neuroscience and EECS. And so I'd be happy to take any questions.
Yeah, I think we have time for 1 or 2 quick questions.
I have two questions. The first is, what is the cost of the hardware itself and the usage for the in-home therapeutic solution in your mind?
No,
Yeah.
repeat the question.
Sure. So the question was, what was the cost for this in-human therapeutic solution that was the muscle invasive brain-machine interface? That's a really good question. I don't know the total number itself, but I can tell you what contributes to the cost. So one is the surgery itself. And I'd say when I think about cost, one is money, of course, but the second is also risk. So I think the invasive surgery is a huge risk. I think there's probably not very many people who are that excited about doing invasive surgery, especially, I'll say this participant was ambulatory. So, she could walk around, she had a perfectly intact other limb, which she used to kind of do most of her activities of daily living. And so to take the risk for invasive surgery just to get back one limb is something I think very few people would do. So there's the surgery itself. And then I think the biggest cost was the personnel. So there were 2 to 3 physical therapists that would rotate for these 4 years every day. And so salaries for them, I think check-ins for, we also had neurologists that would come do clinical scales. We had the neurosurgeon that would come check in on the implant. We had neuroimaging. The robot itself was something that was part of a research study, but is currently, in works of or in thoughts of being commercialized. And the starting cost for this has been proposed to be about 100,000 euros. And so, yeah, I'd say easily upwards of like 1 to 2 million. Yeah. Sorry?
1 to 2 million per person?
Yes. Yeah.
So the second question is like more on the technical side. So it discusses about a very high density array.
Yeah.
Implanted like 300 to 400 like per array.
Yeah.
So do you observe some crosstalk or crossing interference problem? Yeah.
Yeah, that's a really good question. So we have ordered our probes. They've not come yet. But my understanding is actually folks have not. One thing that you can see is evidence of the same neuron on multiple contacts that are nearby, because these contacts are so small, like 20 by 20 microns. But the waveform looks a little bit different. So we know from other people doing neurophysiology studies that depending on whether you're sampling from the dendrites or the axon or the cell body, you're going to see slightly different waveform characteristics, and that's what they see there. So I don't call that crosstalk. I think it's signal. Yeah, it's something that you can use to do better detection of that spiking. So yeah, does that answer your question?
One last question. Okay, let's thank our speaker again.
Thanks.